It's a great pleasure to be able to welcome Loren Looger here as the keynote speaker to this year's conference. My PhD advisor used to say that science is sort of a punctuated equilibrium, to steal a concept from evolution, and that there are technical advances that really drive periods of rapid discovery. And you can look over the past several decades and point to advances in Krelock's technology for labeling and manipulating individual cells, to advances in microscopy such as two-photon imaging, super-resolution imaging, which have allowed us to study molecular events inside individual cells that are alive and behaving, as well as study the signaling between cells and intact circuits in the nervous system, for example. So these are advances that have really been dependent on the skills and creativity of engineers that have allowed biologists like myself to make advances. Loren Looger is one of those very gifted engineers who's really been at the forefront of developing new technologies. So Loren did his undergraduate at Stanford in chemistry and a master's in mathematics, also at Stanford. And in looking at his CV, it was embarrassing. He was actually fairly accomplished before he even went to college, being invited to a number of Olympiads and things like that. From there, he went to Duke, where he did his PhD with Dr. Halinga, developing new probes for assaying the activity of cells and manipulating cells. And that's really been the theme throughout his career. From his PhD at Duke, then he returned back to Stanford and actually went to the Carnegie Institute at Stanford, which is a place that's known for plant biology, of all things. And so he was developing probes to look at signaling and fungus and other exciting topics. But I think he, at some point he was influenced along the way and began to dabble in neuroscience. And from there, he moved to Janelia in 2006, and he's now a group leader there. In addition to his day job at Janelia, he is the founder of 2 companies and holds a number of patents for the probes that he's developed and their uses in a variety of contexts. And I was looking at his CV, and he actually has security level clearance from the Navy. So this is, I think there are probably few people that are better positioned to interrogate the behavior of cells. He has some high level clearance for doing this. Obviously, one of the things that's really, I think, hugely important in what Loren has done was to develop sensors that are proteins. So these are things that can be genetically encoded, they can be expressed in specific cells, and they've overcome many of the limitations that we've had with indicators that have to be loaded into cells, that require detergents, that lack cell specificity, that aren't useful for long-term imaging. So this is all really changed now that we can have access to indicators that are genetically encoded. And so this is the huge advance that Loren has really been at the forefront of. And I think tonight we're going to hear about all the latest and greatest tools. And I have to say, personally, I'm just thankful that I don't have to pay royalties on every Gcamp mouse that we use in our experiments. So please don't start invoking that. But we're looking forward to hearing. Great. Well, thank you so much for coming.
Just sending off a couple emails here. Presenter view. Well, how's the? Okay. That sounded too loud. How does that sound? Does that sound better? Okay. So, who here has heard one of my talks before? Okay. So if you've heard my talk before, you know that the first thing I do is I end up apologizing for something. So it's usually the stuff like, hey, the kid ate my presentation, I'm really tired. If anybody was at McKnight last year, then you remember that what I was apologizing for was I got up to, and I was setting up my laptop, and I turned on my microphone, and I was like, I just got back from a 100-mile bike ride, and I think you guys should probably know that. And I got like 3 slides in, and I was like, oh fuck. so that like 2 different people are like, Pepsi, and they like sprinted out. And so like I pulled up a chair, and I was like, because I was training for this Ironman, which I was supposed to be doing. I was supposed to have done an Ironman last week, but I broke my foot, which would have been my excuse that I was apologizing for, except I'm apologizing about this fucking dog. So I had a shitty day because, I'm here, the wife and kids are at the beach, our best friend is at the beach with them, and so we have a bunch of dogs, we have chickens, we have lizards, we have rats, somebody's got to look after that. So we had the house sitter there and the best friends at the beach, and so their dog, Pinky, right there, is staying with us. And, everything's going well, and then I get a call this morning at 10 a.m. from the house sitter that says, yeah, the dog's been gone for 5 hours. She ate a hole in the fence and dug under. And so I spent my morning talking to animal control. The number on her tag wasn't correct, so I had to make this little fuck. And so I didn't hear anything for 5 hours. And so so I missed Ball's talk, except, you saw me. I walked in, I sat right there, right when he said, oh, and, and thanks to Loren Looger for making the Gcamp. And at that point, I thought the dog was dead. And so you noticed I was sitting right here, and, people were asking questions, and I kept trying to get Ball's attention because I just wanted to go outside and say, dude, I think I'm losing my shit, I need a hug, but between the time that he finished his questions, so he's like, yeah, I'm a little busy right now. I got to introduce the next speaker. And while he was introducing the next speaker, I get the text that says, dog found. And I was like, hey, Ball, we still got to talk. I'm really happy. And he's like, and so then my day got a lot better. But so that's what I'm apologizing for today, is if I seem a little scattered, and if I seem like I should have been working on organizing this talk for about 5 more hours than I did, well, that's who I'm going to blame. So with that, I would like to thank the organizers for inviting me to the, I love this meeting. I love this meeting.
I'm really sorry about the dog.
No. It's fine. It's fine. But I mean, it's usually in September, right? I mean, so okay. Well, all right. Here's my real title. Tools for studying Glia. I think Glia. So at this point you're probably wondering, was that guy drunk already when he showed up? But you're also wondering, why is this guy giving the keynote? I mean, he can't even pronounce the name of the thing that the meeting's on. But I assure you that I am I did the experiment this afternoon, and I'm good. So don't worry, you're in good hands. All right. So now I'll say something, try to get more serious here. So my job, what I've done with this talk is, you guys have a lot of problems, and I'm the guy that fixes problems, right? I mean, I'm like the fixer. You know, I don't give a shit about neurons. I mean, you know, it's just a hard problem, right? I mean, so I come in and I fix their problems. I come in, you guys got problems, I'm gonna try to fix your problems, right? So what we're gonna do during this talk is I'm gonna try to give you tools and ideas for how to figure out what these things are and what they're doing. And I want to set some ground rules here. If any of you have ever heard my talks, you also know that I set ground rules. I want you guys to butt in if you have questions or if you like, this is boring, like fast forward. If anybody was here at my Cold Spring Harbor talk last year at the Synapses one, you'll know that that was the first Choose Your Own Adventure talk I gave, where I just wrote up the topics that people could choose from, and you guys screamed out numbers, and whichever number was loudest, I went to that one. And so we can do that. But, and so you'll notice, like, when we get to the section things, I made them so big that I can actually easily find them from the slide index, so we can bounce around. But here's the 30,000-foot view. I make tools. This is going to go really fucking fast. And, you know, you don't need to try to get every last little thing from this. What I want you to decide at the end of this is whether you want to use any of these tools. And you don't really need to take notes, because if you want my slides, just email me and I'll send you a Dropbox links to my slide. If you want my, obviously published, but if you want my preprint manuscripts, actually like not even submitted yet, just send me, I'll send you a Dropbox links to those. Everything we do is open source, so DNA, everything goes into Addgene. Viruses go into UPenn Vector Core, and yes, our GFAP viruses are selling very well. Good job, guys. Transgenic mice go to Jax, you know, flies go to Bloomington, fish and worms just kind of sit around until, you know, they don't have a standard, you know, way to deal with them, so that's their problem. But bottom line is, you know, almost everything I'm going to show you is, you know, a collaboration. And if you've looked at the author lists on my papers, you're like, how did he even know that many people? You know, I published a 50 author, that's not a genome paper. I mean, you know, like a 50 author, like biology paper. So, you know, we just get all the people that have all the expertise that we need on the team. So get on board if you want. All right, here's your problem. You got these couple of cell types. You know, you got I learned this on Wikipedia. You know, so you've got the major ones up there. Looks like you've got some progenitor cells that sometimes they have funny names.
This is a laser pointer?
Oh yeah, right. Yeah, there we go. Yeah, oh yeah. You know, okay, so you guys, you know, and we heard a lot about this, we've heard a lot about this. I haven't heard so much about these and these, but honestly, I wasn't really paying attention today, so maybe you guys talked about that all day long and I missed it. So we're going to try to figure out what those are doing. This is my problem, right? My problem's really fucking hard too, right? And, you know, to put it bluntly, our problem is probably harder than your problem. And I don't mean that to be offensive, I don't mean it to be condescending, and I don't actually think it's controversial. That, you know, neuronal cell type diversity is probably 3 orders of magnitude higher than your cell type diversity. Right? I mean, you guys may shock us, right? I mean, I don't know, these deep sequencing, maybe you name some new cell types, etc. But our problem is so hard that 200 years ago they realized how hard it was, right? And honestly, you guys are behind, right? No, you're behind. So,
homework assignment, because he's shown that astrocytes are as diverse as the neurons.
well, look, I mean, then that's great, right? I mean, that's great. I mean, it's more problems to solve, right? Look, I don't give a shit which one's harder. I'm saying that, look, there's a lot of similarity between these things. Everybody funds the neurons, and look, we have all the fancy tools. We're doing whole brain imaging. We have a whole $100 billion initiative that I think mentions Glia like 10 times in a 70-page document. So I want to help give you guys the tools you need to catch up, okay? And I'm just trying to help. I'm just trying to help. Okay, so what are these Glia, and how are we going to figure out what they're doing? So, just pretend I'm an alien. It shouldn't be too hard at this point. I've just landed, and I'm very good at biology. Science, let's say. It's very good at science, but I'm a silicon-based life form, and I'm trying to figure out how you guys work. I'll be like, man, okay. First thing is I need to figure out, what do the cells look like? Where are they? Like how big are they? Then we want to figure out what they're doing. They're probably talking to each other. So figure out a little bit about that. 3, we might want to say, okay, let's start taking them apart, seeing what's in there, what's doing what, count things, that sort of stuff. 4 will be like, man, how are they all hooked together into a system, and could this be making people sick, and, maybe we should, take a look in this. And then, to me, 5 and 6, and we're not even going to come close to these today. We're going to do 1 through 4. 5 and 6, I mean, this just blows my mind. I mean, that we work, and we worked at every age, and that we have, wow. Okay, structure. So how are you going to figure out what, you want to figure out where these cells are, how they're talking to each other. I want to sell you a couple tools. So, lots of these things are made first for neurons, and I am trying to sell you guys on repurposing them for astrocytes. Some things, not Glia, I think by the end you'll say, man, he did actually know a little about Glia after all. Because some things we've already shown work. But a lot of these things, I'm going to show you stuff about neurons, and we'll infer that they would probably work pretty darn well for Glia. Anybody who's tried to do, tracing studies knows that the current tools suck. GFP is super, and so you get 1 channel of tracing, and then when you go to your 2nd channel of tracing, you're like, man, I should have picked a different project. TD tomato is horrible. It bleeds into the green. It clumps. It bleaches in cherry clumps and often doesn't even leave the soma. Lots of RFPs make cells swell. The CFPs are really dim and bleached. You can't tell the difference between them. The BFPs are terrible. There are papers where you can do like 2-color with GFP and TD tomato, but if you read the supplementary methods, they had 10,000 times the exposure time on the TD tomato channel that they did on the GFP. That is unacceptable. So I am going to give you a complete replacement of this system that gives you up to 8 identical, mutually orthogonal channels that are each as good or better than GFP. Will change your life. I will try to, so everybody, Rainbow, like New York Times, BBC, etc. Well, we've learned dick from Brainbow, right? I mean, you get the mice from Jack's, they're all red, you know, because they've recombined out, and then it's like different shades of brown. So I want to sell you a way to actually take these things, and we've already done it, to make something that is like 10 times better than Brainbow. And we've actually already done it for Glia. And I'm going to show you the sort of stuff you can pull off. And you can start to think about, you guys don't call them circuits, but I think you probably should pretty soon. You can start to envision reconstructing whole circuits of neurons and glia, morphologically. And then for people that care about molecules, super resolution, et cetera, I want to show you tricks for that. Okay, so these multicolor morphological tracers I speak of, these are called the spaghetti monster fluorescent proteins. So what we've done is we've taken robust fluorescent proteins. We started with superfolder GFP, but we've also made mWasabi and mRuby2 based things. We've deleted the chromophore, so these guys are totally dark. They are just really good scaffolds. And they're super soluble, cells love them, they diffuse, they go into fine dent, everything. And we've jammed them full in an intelligent way with epitopes, all of a single kind. And we think that it looks like a fuzzy, fuzzy ball, so we call it the spaghetti epitopes. First we demonstrated it in flies, so if you ever see something from Jerry Rubin and he's like, here's our Gal4s, these are all spaghetti monsters instead of GFP. You just see green and you assume GFP. He ditched GFP 5 years ago and replaced everything with the spaghetti monsters because the antigenic labeling is so much more that he found that the labeling that he gets with primary antibody on a spaghetti is more than he gets with primary and secondary labeling on a GFP. So these things are bright and they're, boom, they fill the brain. This is now in mouse. It's useless, tells you nothing. But they're really good at tracing very thin little things. It's more obvious here in the next slide, where what we've done is we've injected 2 flavors of these. One is flag epitope flavor, the other is Mick epitope flavor, into the 2 hemispheres of whisker somatosensory cortex. And you can see, and then one we've labeled, so this is all immunohistochemistry. These guys are not intrinsically bright. We've labeled this guy with the red labeled antibody and this guy with the green labeled antibody. You can see we get beautiful labeling of the injection sites, but hey, look, we got beautiful, you can see the axons crossing in the commissure, and this is way, this density is so good that it's actually good enough for good computer algorithms to trace. So NeuroLucida loves our stuff, whereas that usually anything less than like a Golgi fill or a Biocitin or whatever, it's like nope, and gives you a bunch of fragments. It loves reconstructing these things, and you can see here we went down to thalamic afferents, and you can see some beautiful terminals, axons, so these things are great. We have up to 8 orthogonal colors of these things. We could give them to you as straight virus, flexed virus, so Cree-dependent virus. You could put them in your astrocytes, whatever you want. Can we maybe turn down the lights a little bit more on the screen? Awesome. Okay, so what we've done here is we have gotten the same amount of DNA encoding GFP and then one of our spaghetti monsters into CA3 hippocampus, and then we've done immunohistochemistry with exactly identical parameters. the antibodies, the imaging, exposure time, blah, blah, blah. You can see we're filling the processes way better. You zoom in, we're filling spine heads way better. And then, have you guys heard of this awesome synapse called the thorny excrescence? It's the dentate gyrus synapse in the hippocampus. Cahal noticed it and was like, whoa, this is the craziest thing I've ever seen. It is 16 to 20 little spines stacked on top of each other in this weird configuration. You can see that is a thorny excrescence spine. So Cahal got it by Golgi staining. The only other way it was ever visualized was in electron microscopy, because you could not, you express GFP and you just don't get enough label. The only way that people did finally see it in fluorescence was you inject Lucifer yellow, fix, do antibodies against Lucifer yellow, and then secondary. So these things are highly antigenic. This is just a head-to-head comparison of a spaghetti versus a TD tomato as your second color. So, I mean, if I were you, I would just throw out your TD tomato virus. I mean, honestly, I would throw out your TD tomato virus. The antibodies are terrible. Imaging them directly is terrible. I mean, just everything is terrible about it. So just throw those out and replace everything with, you know, like nice second channel there, nice. So they're really useful in high resolution imaging as well. This is CA1 hippocampus. This is in array tomography. I don't know if you guys have seen this. It's a really cool technique where you thin cut so you Z-section everything to really thin, so you get great Z density, and it improves your XY density, of course. Then you make a big ribbon out of everything, and you can keep reprobing it. So these things are the best existing antigens for these ultra-thin sections for this high-resolution imaging. And so, you know, you can count synapses with this. They are counting synapses of various cellular classes. And array tomography is just getting easier and easier. They're so antigenic that they work great in immuno-EM. You know, anybody who's ever looked at an immuno-EM paper, you know, usually it's like kind of comic relief. You see like one dot that they've pointed an arrow to, and then like 10 dots over here, and then like something they photoshopped out. But I want you to notice, like, that, you know, that this is a dendrite, and that has 1,000 gold nanoparticles on it. And this is a smaller dendrite with probably 500 gold nanoparticles. And, you know, so these are gold. This is like 6 nanometer. This is a 12 nanometer. This was good enough to do easily a 50 serial reconstruction of these entwined dendritic arbors with, you know, pretty easy. I mean, and this is, I mean, fail safe. I mean, you know, my kid could say, yep, there's the label, you know, then, you know. So so what I've shown you now is just tracing large sets of things. How about if you want to go back, you know, hundreds of years to what Golgi was doing, of like individual cells. So, you know, there's lots of things. Jerry at Janelia is getting really good at this flip out, where you have a temperature-sensitive promoter that you'll a heat shock promoter rather, and you'll stochastically recombine it out, and you'll only get your thing in one cell. And so you get these beautiful. I mean, this really goes back to like Golgi versus Cahal. I mean, of like, you know, being able to see every last bit of single cells. But where it gets super cool is, so I told you that there's the 8 orthogonal channels of these super antigens, each of which you can bring on either primaries and then labeled secondaries, or even just directly labeled primaries. Now, since these are Alexas, the bandwidths are very tiny. So you can actually fit 8 in the spectrum with no overlap, unlike Rainbow, where you have the 3 and they're kind of all over each other, because fluorescent protein spectra are really broad. So what you do is, I think this is 5 epitopes, and then instead of Cre, because Cre is toxic to flies, they use this other recombinase flip that stochastically gives you different combinations, and then you just do immunohistochemistry, and you can see you pretty much have a tiling of every epithelial glia in the Drosophila lamina, part of the early visual system. These are high-resolution images. You zoom in, you can start to see, and if you had like some expressed in the neurons, you could start to, count contacts, be pretty quantitative. So this is gliabo. That was with antigens. There's another way to figure out where your cells are, and that is make better fluorescent proteins. Yeah, Mark.
So maybe you're about to talk about this,
Yeah.
but so repeat this so everyone can hear it.
Yeah.
That all depends on fixing and staining, right?
Yep.
So
Yeah.
that's one limitation.
Yeah, but honestly, when you really drill deep into what people want to do with Rainbow, the only person who's really interested in live Rainbow is Jeff Lichtman. Because his problem has to be in vivo, because he's looking at those 2 axons that are coming and they're competing and one's going to win, and you can't do that on fixed. He's got to see the dynamics. Everyone else wants to count cells, see how many cells of what shape, who's bumping into whom. Everyone else really wants to do it in fixed tissue. And so, his new paper, he calls it anti-bow, basically like this, but shittier. Because he's still using the fluorescent proteins as the antigens themselves, and they're not terribly good antigens, right? So we took the good things about the fluorescent proteins, but then built in the best antigen antibody combinations to make everything. And so the other reason why it's good for this is, oops, wrong way. Because all of our guys are built on single scaffolds with just the epitopes changing, they traffic similarly. If you dig into the raw data of Rainbow, you see that each of the fluorescent proteins localizes to different subcellular regions. And so that brown that you get is like it's a computationally merged brown, because the red was in like vacuoles and the green was membrane associated. And it's a headache. Here, everything migrates completely, is trafficked completely similar with same scaffold. So that's why you get beautiful, these are unprocessed. That is not photoshopped. That is right off the microscope.
Have you guys put spaghetti monster? I can't believe I'm saying a sentence. Have you put spaghetti monster on an antibody for flow cytometry?
Put it on antibody for what?
To do flow cytometry? Like you sort cells.
Oh, no, we'll get to that. Yeah, so I can solve your problems. So, yeah, I can solve that. I'm not going to stick it on an antibody, though. I'm going to stick it somewhere else, but you're going to love it. So the other way you can do it is, with fluorescent proteins. You look, fluorescent proteins in my bread and butter, right? So, but one thing that they don't do well is they don't do electron microscopy well, because you guys know, you work with GFP, if you put a little too much PFA or a little too much glut on there, you get these clumps, you get these weird autofluorescent hazes. If you even think about putting osmium on there, forget it. It's done. So what we did is we said, hey, wouldn't it be cool to have a protein to do like palm super resolution imaging that you could just hand it to someone and say, express this in your brain, and then do EM like you normally do. And it works. So what we had to do is we had to use rational design. So you look at EOS and you think about what PFA and GLUT and osmium are going to do. They love nucleophilic residues. There's a cysteine, a stupid cysteine on EOS. That's not going to help, right? So we got rid of all these things. We did hundreds and thousands of these guys. We made a little 96-well plate assay of like making them, squirting osmium on them, screening, adding, et cetera, et cetera. I don't want to bore you with it, but what we ended up with is, it didn't really come out very well, but these are mitochondria in 323 cells, and this is MEOS2, and we made two guys, MEOS4A and MEOS4B. This is no osmium, so that just means like 4% PFA and I think 0.2% GLUT. Boom, they're awesome. Awesome. You're counting your mitos. 1% osmium, not awesome, right? There's nothing. There's absolutely nothing. No fluorescent protein until these guys survived 1% osmium is like a nuclear bomb going off in your sample. And so these guys, I mean, they're not 100% fluorescent as they were before you added the osmium, but they're 40% as fluorescent as they were, which is pretty damn good. So, it's just not projecting well, but these are actually really nice, nicely filled mitos. And this lets you do really cool stuff. So this is now, again, a 3T3 cell. We have targeted, this is MEOS4A. We've targeted it to the mitochondrial lumen, and then we've hit it with a primary fix of PFA and GLUT, and I think this is then direct chemical fixation. You just go right on top with the osmium, and then you infiltrate the GMA. And so we have two ways of doing it. One is consecutive sections where you're serial slicing it. And that's what this is. So you serial slice it, and one guy goes onto a palm grid and one guy goes onto an EM grid, but they've all been through this horrible witch's brew. Sometimes they have 1% uranyl acetate. That can come on as a post stain after you've got them cut. Lead, tyramide amplification, and it's still brightly fluorescent. It's still so fluorescent that you get enough photons out to get really great super resolution imaging. So that was TEM. This is exactly the same. So this is same section. Sorry. The other one will be, this is same section. So we cut a slice, put it on an EM grid, did the fluorescent imaging, did TEM, and then did SEM, all on the same sample. And that's TEM in palm overlaid, and that's SEM in palm overlaid. And this shows you how good the resolution is. You can see the ultrastructure. You get beautiful cristae, which you never could, like in attempts to do palm, clem. You're lucky if you have membranes. You don't have cristae, you don't have bilayers, you got very little. So we were really happy with our cristae in the EM, and actually if you stare, the cristae line up perfectly with the gaps in the EOS density. So I think it really is telling you a lot about the shape of the innards of the mitochondria and where the matrix and the lumen and the cristae are talking to each other. And this, again, they're not projecting well. I just did screen grabs. I think I lost a lot of resolution that way. I'm sorry. But there was a beautiful thing of rough endoplasmic reticulum. You could count the ribosomes stacked on there. And over here, you can see both sides of the nuclear bilayer. So, I mean, this 1% osmium, this is GMA. GMA is not the perfect resin, which leads us to, we're just getting it to work in epoxy resins. EPON is the gold standard. That's what the brain people won't touch it until it's working in EPON, because it's so hard that you can cut thin slices with 0% loss. GMA has to have a little water in it, and so like 0.1% of your slices get kind of squished. And that sounds great, 99.9, but when you're reconstructing like dendrites and axons, it's actually super bad. So trying to get it to work in these epoxy resins. So, and we'll come back to this. This is not done yet. We're going to talk about this in parts list 2. But now let's figure out, how do we figure out what these Glia are doing?
Lauren,
Yeah. Talk to me.
tag proteins that have been fine with GFP, with Spaghetti Monster, and seen that they traffic the same and everything looks good?
Yeah, so I'm going to show you that one of the EOSes, MEOS 4B, is as monomeric and fusible as GFP, and that all the Spaghetti Monsters are as monomeric and fusible as GFP. We're doing protein traps. We're doing, just direct fusions. MEOS 4B and GFP scored miles ahead above all the other guys, all the other M proteins that are really like D or T, monomeric, dimeric, tetrameric. When you make a new protein, fluorescent protein, you have to put M in front of it, right? But I mean, even some of them are obligate dimers, and it's still, it's M plum, So it's monomeric. No. So we're going to pick up speed a little bit here. You guys know a lot about Gcamp, right? So we're gonna pick up some speed here. So Gcamp is great for instantaneously seeing what's going on in cells. Got some electrodes in there. They're spiking, and when they spike, they turn green. Big deal. We wanted to make these guys much better, so we invented this automated robots. We've got, we're producing, hundreds of thousands of lentiviral particle, wells of lentiviral particles of different variants, infecting primary neurons, plating them out, dropping in a 96-way well of electrodes, zapping them with platinum wires, measuring everything on a CCD, and then quantifying it. We can give you plans if you want to build one of these, but I wouldn't recommend it. This is how it works. The movie broke, but you zap it, and you get a little transient. And then you just keep making mutation after mutation. And so these are the things that we screen to get to the Gcamp6 mutants. These sorts of plots, you're going to see what we have up here at the top is the sensitivity, the one action potential response. Then comes a heat map of dynamic range across all the stimuli. Then comes kinetics, where lower is faster. And then comes resting fluorescence. So depending on who you ask, some people want your Gcamp to be bright at rest, and other people want it to be dim. Particularly the fly people want it to be bright. because you're imaging almost exclusively in neuropil, and they're having trouble finding it if it's not dim, and they're having trouble co-expressing. Whereas the mouse people, it's easier to find your region, and so we really think, so Gcamp 7s, we're really going to drop the resting fluorescence. It'll be near 0. And you should blow the SNR out the roof. But you can see, so we screened thousands of these guys. This is published. I'm just going to rush through this. They're really good. You get these beautiful 1AP responses. They hold pretty good dynamic range. They work beautiful in vivo. This is Gcamp 5G. This is visual cortex looking at direction selective cells. This is the best Gcamp we had before. Gcamp 6S labels pretty much so 60% of cells are labeled as orientation selective in V1, which pretty much matches electrical recordings. OGB has been mentioned a lot. So the problems with OGB that were mentioned was it's hard to load and you only get a couple hours. The other problem with OGB, you're missing a lot of shit. You only get, so OGB annotates 35% of V1 as being direction selective. Gcamp 6S annotates something like 55%. So not only was OGB a pain, it was not the 100%, it was not the ground truth that we thought it was. So OGB had tons of false negatives, which is bad. And so again, you guys have seen 6F, we're getting them faster. If you read the paper, you can see that we get these beautiful single spine responses. You can find out that individual spines have orientation selectivity. The diversity of orientation tuning of spines turns out to be way more diverse than the tuning of the cells that they compose. So you've got some straight direction, you got some orientation broad, you got some orientation narrow. We could keep measuring this over a couple of weeks. Most guys were pretty stable. Most spines were pretty stable. A couple of them drifted. And we actually think it was pretty interesting. We could see what was going on when they were drifting. And then the coup de grace that I don't think is done yet, and I think people like Ling Yi are going to help us really crush this problem. So we did, 20 laboriously, 20 two-photon tiles, and just keep reproducing the stimulus. I mean, it's a really reproducible stimulus. You show the gratings, the spines and cells fire. So we managed to assemble something like 500 spine maps, and then obviously we can do the soma. And then we showed that, comfortingly, the soma is the average of the 500 spines that we did. So that's kind of cool. But this pyramidal cell doesn't have 500 spines. It probably has 20,000 spines. So, now I want to figure out a way. And again, I think it's, optics pioneers that are going to help us. I want to do every single spine. I think there's some more secrets hiding in there. But again, okay, so here's the fireworks, brain initiative. I mean, this is, so I think no matter what happens with the brain initiative, I think it will be declared a success, largely on the basis of the science that existed before it began. Right, because, so I mean, this is amazing. So this is a 4 DPF larval zebrafish embryo. It's pan-neuronally expressing Gcamp 6S. If you look really close and are a morphological expert, you'll notice that the 6S looks a little weird. I've fused it to a histone. So this is H2B Gcamp 6S, because that way it got rid of our neuropil. This is like autofluorescent haze rather than neuropil. signal. And let us get our, because now, you know, when the cells are touching, you have this whole, you know, it's diffraction limited, so everybody bleeds into everybody else, and you can't get perfect cellular resolution. When it's attached to the chromatin, we get something like 95% cellular resolution of these responses. You may say, well, you're measuring nuclear calcium. What does that mean? I don't know. But it's correlated. We show that it is correlated with spikes. We get a pretty linear tuning curve of sticking an electrode in one of these cells, firing spikes, and getting responses from our H2B Gcamp 6S. And this fish is behaving. It's not just chilling out. It is paralyzed. It is in a virtual reality environment. We are showing it these, on the bottom is an LCD projector that's showing it these moving gratings. We have 2 electrodes in the spinal cord that even though it's paralyzed, the motor neurons fire, and so you can tell the fish's intended motion, and you use that to create a closed loop. So and then you can fuck with his physics. So you do things like you change the gain up 3x, and then he's like, oh man, like I know I'm flying. And then you drop the gain, and he's like, damn, I'm getting fat, you know. And so I mean, this is what we can do. I mean, you know, like if you had said 5 years ago that we will be measuring every single action potential in about 95% of a complete vertebrate brain while it is doing a fairly complex behavior, I think you would have gotten laughed off the stage. And no, I mean, this is the image they use for the Brain Initiative. You know, if you look at the press releases, this is the picture at the top. And so you can go further. So, you know, we started to, so this is made by we, I mean, I did Dick, and Misha Ahrens and Jeremy Freeman and Philip Keller and the postdocs did things, you know, starting to do optomotor, optokinetic, all these, you know, classical behaviors. And so you can start to assign so this is orientation selectivity, but you can assign all these profiles to every single cell in the brain, and you find that you know, if you say, oh, where in the brain are direction selective cells? I mean, so it's not a rodent, so you wouldn't say there's no cortex. We have tectum, which is colliculus, which is this junk here. But the whole bloody brain is direction selective. I don't know what this means. Astrocytes. So, you know, these are, so Gcamps, you know, aren't just for neurons. So this is a cool collaboration with Erlen Nagelhuis, who unfortunately couldn't make it here, Rolf Sprengel, and then Wanan Tong. What they're doing is, and again, nothing is projecting well, but they're injecting current into the Schaefer collaterals and doing 2-photon frame scans of Gcamp. Either this is cytoplasmic or Ball's magic membrane luck tethered version. And you know, so you can see we're getting more somatic response, you know, beautiful somatic response there with the cytoplasmic one, and then I couldn't figure out how to loop in PowerPoint. And then you'll see you're getting, you know, way more, you know, there was a long thing right there. And so hey, these things are great for detecting, and you guys saw this morning in the session if I, you know, had been paying the remotest bit of attention. Those were slice. Those were slice. It also works in vivo, and this is the work of this amazing man sitting right there, Joe Sichon. We're going to talk more about him later. So he's in Wen Biao's lab, And what usually happens is I develop a new tool, I send it up to him. 2 weeks later, he sends me a draft manuscript of some finding. So so what happens? I send him these Gcamp 6s. They've just got a CAG promoter, so we're getting all the cells. You'll notice that we're getting some neurons, and then we're getting some astros. So this is layer 5, motor cortex, and here the mouse is just it's chilling. So that was so the neurons are more recognizable here. Here's some astrocyte somas going off. Now you turn the treadmill on. Now this guy's got to run, and you see that the overall level. So now you got motion artifacts. But the overall level of activity goes up too, and you can start to really tweeze things apart. There's this beautiful dendrite with a ton of, it's probably a dendritic calcium spike being fired there. But you're getting these beautiful responses from astrocyte somas there, some beautiful astrocyte end feet. This is back up in layer 1. Where lots of the astrocyte end feet are. What's something else we can do with Gcamps that's really cool? Tag them to things. And this is something that Mark and I have talked about for years. I know he's put a lot of energy into it. We've put a lot of energy into it. Hope we can all, get it all organized. We have shown the Gcamps work well when, and others. Ball stuck it at the membrane, Leon Lignato stuck it at the presynapse. We stuck it to nucleus and others stuck it also. Lots of stuff you can do there. What's coming with Gcamp 7s? They're going to be even faster. We're going to have a wide range of affinities. The mouse people love Gcamp 6. The fly people hate it. They're like, it's way too high affinity. We need, our neurons are spiking at like 200 hertz, and then they'll modulate up to like 230 hertz. And your 100 nanomolar sensor's not really going to cut it in that range. So we've pushed the affinity, like 4 logs out. And so we've got these guys that are responding much better for really fast spiking neurons, where you're now not like counting spike times, you're measuring spike rate. So the fly people at Janelia are loving this, and these will be out soon. And again, for the mouse, the direction things seem to be heading in is the mouse people are greedy, and they want more and more, they want more to be more like the fish. And so, but they get that by zooming out, but right now when you zoom out, you lose too many photons. So what they want to do is they want to drop that resting fluorescence, which the fly people want to find your region of interest, they want to drop that to 0. So then when you do the math for signal to noise ratio, it's through the roof. And now you're getting, now you can use like a 10x objective and have like 50,000 neurons in your field of view. Probably still get cellular resolution and single action potential sensitivity. The last thing I want to tell you about Gcamps is that this is going to make your life so much easier. The Transgenics are on their way. I'm sorry I wasted everyone's time with the Gcamp 3 transgenic. It just wasn't bright enough or sensitive enough. The 6s are the real deal. These are some Thai-1s that we made at Janelia. That probably doesn't interest anybody in this audience unless you wanted to use Thai-1 neuron in something else, in an astrocyte or something like that. But in collaboration with Alan, we made a pre-dependent versions of 6S and F that should ship from JAX pretty soon. So that should get you guys in business. Then you get early development. Then you don't have to have this other survival surgery. the viral, after, 2 months, your cells get Gcamped out. Whereas with the transgenic, that never happens. That was green. You want things for red, so you can do 2-color combined with optogenetics, less autofluorescence, and then real pioneers like Chris Hsu keep doing what should theoretically be impossible, of like going deeper and deeper and longer, and we'll touch on this. I think I can solve Wen Biao's problem of imaging through a monkey pia. I think I can get him a laser where he can image microglia at 3 millimeters under an intact pia in a macaque monkey. He'll have to do a little craniotomy, but he can leave the pia intact. So just very briefly, how do you make a red one? Well, duh, you get rid of the GFP and you put in the RFP. It's that easy. They work. They're spectrally separable from the green, so you can use them for 2-color. We solved the crystal structures. That helped us make them way better. That's old news. Another one of these plots, these are RCamps. So that's the one we invented. Our gecko is the one that Robert Campbell invented. There's pros and cons. I think they're not really competitive tools. I think they're orthogonal tools, and we'll get into the reason why. This line right here is where RCamp won. I think it's that one right there. So they're, 10, and this slide is like 4 months old. They're at least 10 times better. I think they're right up here now. So like a Gcamp 5.5 level. And they've also gotten a lot brighter, they've gotten a lot faster without losing brightness. Similar with the R geckos. The R geckos are now more sensitive than Gcamp 6S. So this is now the most sensitive gecko on the market. So again, I told you they're sensitive, some fast, some of them are very photostable. And again, we realize not everyone, and particularly maybe people like you don't want super high affinity things for pulling out every action potential in auditory cortex that's spiking at 0.05 hertz. I mean, you want like a more broader dynamic range. So we're making these super low affinity versions, that could be, maybe Astros need them, ER, Mito, etc. Let me leave you with 2 thoughts. One, so our gecko is currently a bit more sensitive than RCamp, but it comes at a price. And that is this photo switching artifact. That if you put our gecko or RCamp in a postsynaptic neuron, you put channelrhodopsin in a presynaptic neuron, easy enough. Shine blue light on the presynaptic neuron, you release some glutamate, you spike this guy, calcium flux, measure that. These 2 curves are with and without the channelrhodopsin. So the channelrhodopsin is not important for the response of the R gecko. It's just this photoactivation artifact. It had nothing to do with calcium. So you need to be really careful, and we can't make that better. So I think when you use our gecko, you may have to computationally deal with this. So caveat emptor. So going deep in vivo. Wen Biao is the pioneer of skull thinning. He was complaining to me at the bar. He's like, but it won't work in monkey because of this damn PIA. We've sent all our stuff to Chris Hsu, and in a week he rips out 2, 3, and 4 photon spectra, and has actually shown, he's done microglia imaging at something like 2 millimeters. That's in a rat or something. But I think it will work for monkey. All right, how we doing?
Keep going.
Okay, people are having a good time. Is anybody not having a good time? And
They can leave.
okay, all right. All right. We'll try. We'll be more efficient here. So calcium is not the only interesting ligand, right? So wouldn't it be nice to measure things like neurotransmitters? So we came up with the idea, it's pretty obvious, it's like, hey, let's make a Gcamp that's just for glutamate. So, we did the same thing. You take circular permuted GFP, and instead of calmodulin M13, you rip that off, you stick it on a bacterial glutamate binding protein. For this application, you stick it on the membrane, although I'll show you cytoplasmic glutamate sensing too. And again, here's where, so Joe did almost all this work. Let me, briefly run through. It turned out that the affinity of our first guy was about 4 micromolar, which turns out to be pretty perfect for rodent-like pyramidal cell imaging. But again, for all sorts of other animals and for all sorts of other cell types, like for cerebellar granule cells, that's a bad affinity. And, I gave it to my cerebellum guy, and he's like, man, your shit sucks. There's just no signal at all. It's just really bright, and nothing ever changes. And I was like, well, man, what do you know about cerebellar granule cells? He's like, well, glutamate release rates are something like 1,000 vesicles a second. And I was like, okay, that's not going to work. That's totally saturated, So we had to knock down the affinity by like 100-fold. And now he's getting decent responses. And the same thing with the fly, although the fly, their glutamate is choline, and their choline is, fly people. So it only recognizes glutamate and aspartate just barely. And it is completely orthogonal to every drug you would love to put on there. And this is going to be critically important. And we'll come back to this a lot. This thing is fast. We did it with stop flow fluorescence, and it's essential. This is a diffusion limited on, and then we sent it to this awesome guy, Ed Chapman, and what he does is he doesn't use it as like a virus or a gene. We send him pure protein, and he squirts it on his cells, and then induces exocytosis and endocytosis. And he can do this really quickly. And he says these guys are probably 10x faster than you even thought. Because, when we did our on, they were all flat lines because we had a 1 millisecond dead time on the instrument. So that's why it's just a lower bound. We can, in work I'm not going to show you, we can detect minis. We get these quantal responses. This isn't a special synapse. This is the bipolar cell, retinal bipolar cell. It's a ribbon synapse. It's lots, big glutamate. But I really do think that we can make them sensitive enough to get every single vesicle, even at central synapses. And okay, here's what, so I sent it to Joe. 2 weeks later, I get this. He's injected it into layer 5. And then it, of course, fills up in the layer 1. He has the mouse running on the treadmill. We're imaging in forelimb motor cortex. And he has the mouse is forced to run forward or backward under the user control. And at rest, and so we're measuring up here. And so we can, the responses are limited to little spine heads. At rest, it's just flat. Then when it starts running, you get these huge spikes. When it rests, it's flat again. So it really seems to be, focused to running. Not only that, it's different synapses are tuned to different directions of running. I mean, this has to be the case, right? I mean, if you're going to build a brain that's going to know, how to control, I mean, it had to be this answer, right? But it's cool to see it. It's cool to see answers that had to be that. So these two spines loved reverse. None of them liked resting, and the other guy liked forward running. And when you computationally subtract the images, the signals are just at spine heads. And all the obvious controls work. You add on TTX, you don't get any glue sniffer signal. You add on things that induce seizures, and you get hella glue sniffer signal. Yeah, we did cover the RCamp. So now you can start to play with these things. So this was Corey Bargman's cool idea. We stick RCamp and glue sniffer in the same neuron in the worm. It's in the nerve ring. It's the master interneuron that decides whether or not to back up. And so fortunately, RCamp localizes to the soma. Glue sniffer localizes to the processes that in this particular neuron, and this has been meticulously worked out, are receiving glutamate, and there is no glutamatergic output from this cell. It outputs a neuropeptide like 0.5 millimeter away down onto a muscle. So all we're seeing is presynaptic glutamate. And so, you know, what's coming in is this thing is the green signal. What's coming out is the red signal. And we found we could just make a little leaky integrator model that said, well, let's just assume glutamate is the only thing coming in. Let's leak a little bit just to allow for, you know, model the channels and the, you know, whatever. So I think the leak constant was like 11 per second. And then, so blue becomes our prediction, our fit to the output, which is pretty darn good. It's not perfect because there's other stuff than glutamatergic input to this cell. And fortunately, if you knock out the glutamate transporter, you get no glue sniffer signal. Occasionally, you get these random big spikes. We've solved a lot of mysteries with this. We solved the mystery of the subunit nonlinearity in the retinal ganglion cell. It is the bipolar cell terminal. GABA from starburst amacrine cells is the sole source of direction selectivity. And this study showed us something that was not horribly unexpected, but horrifically alarming for people doing voltage clamp. If you're voltage clamping your soma and you have a thin little dendrite, I can assure you it is not voltage clamped at the same voltage as your soma. And we directly showed this by measuring the glutamate input. We showed that the electrode was lying to us, but the glutamate sensor was telling the truth. So, I mean, there had been warnings, there had been shots over the bow about, you know, voltage clamping neurons with really thin dendrites. You guys don't have this problem, I guess, with your in-feed. I guess you could patch an in-foot, I don't know.
How about sensors for other neurotransmitters?
Give me like 2 slides. Yeah, so, who cares about Keynote? You know, this is Ball's study, and so I want to give a big shout out to Bart Borhaus, with whom I do all my retina work, and a huge shout out to Ball to getting me interested in, you know, astros and other cool cells as well. So I want to say we independently extended Ball's result of glutamate transporters regulating astrocyte glia engagement into circuits with the Mueller glia in the retina. So we expressed glue sniffer along the Mueller glia. It goes all the way up there. And then when we showed, we're giving it these optical, you know, these visual stimuli, nothing's reaching the Mueller glia. But the bipolar cells are releasing, the sensor's working, it's releasing onto the retinal ganglion cell dendrites. When you add TBOA, you block these glutes, now you're getting hella glutamate onto the Mueller glia, and now they're engaging. And it alters the engagement of the retinal ganglion cells. What else would you do with this? Again, we have an affinity series. You want to co-express a reference protein, change color to use with other signals, and target it. Other targets. Okay, so green are things that are working. No one gives a shit. Bold are things that are published, but no one cares about maltose or organophosphate. Glucose, people care a lot about glucose, and I think you may want this. Yellow is almost working, and that includes GABA, acetylcholine, and ATP. Acetylcholine is being done in collaboration with Henry Lester and his great postdoc, Amol. ATP and adenosine and glycine and deserine are being done in collaboration with Ball and his great postdoc, Mark Lobus. And we're halfway to an L-DOPA sensor that hopefully we can then diversify. And then these guys are doable. This is almost impossible, and that's doable. This just says GABA is almost working, 200% increase. Affinity series nicely displays. There's no naturally evolved soluble dopamine binding protein because it's not environmentally abundant, so we had to computationally design one. So we turned an arginine binding protein by computational design. This is like a 20-fold mutant. We turned it into a moderately crappy L-DOPA sensor, but that still works on the surface of neurons. You can add L-DOPA and it blinks. So now we just need to get rid of that, and then we have that, and then we have dopamine. Okay, this is really cool. So what I've shown you so far are instantaneous indicators of, if you want to figure out what's going on in this part of the brain, let's say you want to figure out what's, controlling sleep. You have to know where to point your microscope, right? Because if a neuron blinks in the forest and a microscope is not there to see it, then it didn't happen, right? And so you have to know where to look, and you have to be there, and it often has to be head fixed, and et cetera. So we wanted a Gcamp that remembered what it saw and would tell you later, where you didn't have to worry about head fixation. So now we've got these neurons. So wouldn't it be cool? So Gcamp goes from dim, calcium comes, it's bright, calcium leaves, it's dim. We wanted something that would start with one color, calcium comes, user supplies some stimulus, and something happens, and it changes color. And I'm going to try to convince you this is like a CFOS that works. So the idea is, because it was blindingly obvious, we're good at Gcamp and we're good at EOS. Let's make a Gcamp out of EOS. I mean, you guys are all protein designers enough to design that. So we did that. And so the idea is, hey, here's a bunch of cells. They're all green. Nothing's happened yet. We turn on our photoconverting light. They're firing some spikes. And the more spikes they fire, the more red they get. And so this just says we did it. Took like 2 years of beating to get these mutations, but it works. You get a 20 or 30-fold increase in red to green photoconversion ratio in the presence versus the absence of calcium. Both the green and the red form are negative calcium indicators. This has to be the case from photophysics, and we can talk about it at the bar. We named it Compari because, one, it's obviously a calcium-modulated photoactivatable ratiometric indicator. And then the emission spectrum of Compari is exactly the same as our protein. And only later did I find that there's a Compari tomato that goes from the same green to the same red. So, I mean, it was meant to be. So this just shows that it works in cell culture. What we're doing is we've got some cultured neurons. They're all green. We shine some photostimulating light. Nothing happens. There's no red. We now do it again and zap the heck out of it, 80 hertz. And now, hey, they got really red and they got a little less green. And so what you do is you merge these guys, and you actually take, there you go, there goes the merge. You do the green to red ratio, and you actually get a pretty big dynamic range. And we can take this even further up to like 20-fold, and it's pretty darn linear. So that's in cells. No one cares about cells. Does it work in vivo? It does, and this starts to highlight some really cool aspects of this. So, we figured, hey, we'll start with the most optically transparent sample known to man, the larval zebrafish, pan-neuronally expressed Compari. This guy has been, he's been swimming around a tank. So he's freely swimming because there's no microscope. So he's not head fixed. He's not in a little capillary. He's freely swimming. And we shine a laser pointer at him and annoy him, and he swims away from that. This guy has been trichane anesthetized and is just chilling. So you zoom in. There's no photo conversion in the guy that's just chilling. But the guy that's been pissed at you for chasing him with the laser pointer, you're getting the tons of these red converted cells. And it's nice to see that you get red and green and yellow, and they're all like beautifully opposed to each other. So I think this is real and not just, light penetration. Go back to the hindbrain. The guy that was chilling, he wasn't going to get off the couch. So, the premotor, pre-spinal, there's nothing going on. That's autofluorescence. Whereas this guy, he was swimming. And so you get the spinal cord lights up. You get the premotor, the motor neurons going in there. Fish was easy. Mouse was a little harder. But we made it work. And the reason we made it work is with fish, I don't know what these neurons are. And it's going to take me a long time to figure out what these neurons are. We don't have the genetics. We don't have the markers. We don't even have a good genome. How am I going to figure out what that neuron is? But in mouse, it's easy. We just go back to our favorite, show the moving gratings. And so what we did is, so this is just a 2-photon stack showing you get beautiful expression. What we did, and this is showing the moving gratings while zapping it with the photoconverting light. You get some red stuff and some green stuff. Cool. But how do we know we're right? Well, we use the fact that I told you was a feature in a bug. They're negative indicators. So first we image it with the negative indicators, and so we get the tuning profile of every cell in our field of view so we know the ground truth. Then we repeat it with the photoconversion, and that just says we get it's robust population. You get like a 2 to 3-fold boost. Now, after we've done that trial, you perfuse the animal, you pop out the brain, you do a series of confocal stacks on the entire V1. It turned out to be pretty easy from local landmarks to align this was 2-photon in vivo. We used the vasculature and the cells. We didn't have to warp it too bad. The preservation was pretty good, so it was pretty linear transform. And now we have the orientation selectivity of about 500,000 cells in V1. So now you can start to ask, real population level neuroscience questions about what are the distribution, all these things we've been wanting to know for years. Yeah.
Are there any issues with the depth of the cells in the photo?
Yeah, sure. So UV light sucks. So that.
Was that sample, depth?
These are layer 2, 3, so 250 microns. It's just like getting channeled opsin light in. It's doable, but kind of a pain. Obviously we'd love to redshift this converting light. I don't know. Unless this talk is going to be like 5 hours long, I'm not going to show you any fly data, but it's awesome. Because, the cuticle's hard to get through, but it's so small that, so I'll just give you the punchline. They show the fly the odor, they zap it, and you get a labeled line of olfactory receptor neuron, projection neuron, something to the mushroom body, something to the lateral horn, something to a part of the brain that has no name, and then a descending interneuron down the ventral nerve cord. And you can do it with geosmin that only activates a single glomerulus, and you get one structure lit up in your brain. And you can actually go and trace the red to a terminal, and there's a red spine. And then you trace that to a red terminal, and there's a red spine. How long should I keep talking?
5 minutes. 10 minutes.
Okay. I mean, I can keep talking at the pub or something. I mean, okay, so let's, all right.
Why don't we take a poll?
Okay, yeah, let's see. So what we got here? Hey, wait. So you, yeah, I gotta hook you up. Ooh, ask me in 5 years. Okay. So protein tagging. So, yep, the spaghetti monster is great for tagging it. So this is a cadherin. The other colors are just filling neurons. This is the channel for the cadherin. This is with a spaghetti fused. This is with a single HA epitope. So the spaghettis work well. The EOSs work well. This is lamin A. So, you get this beautiful membrane bilayer. You can fuse it to any protein you like. These guys are as monomeric as GFP and DRAMPA. So, Johanna, I think that should help solve, well, I don't know where you are, but there you are. Hopefully, I think we can fuse this to TFAM finally. I know that wasn't your project, but I'm really excited. So they're super monomeric. The super antigens, I think, can, so we have this project at Janelia called NeuroSeq, which is basically just in fly and mouse, grabbing every single cell class that we have some sort of genetic tag for and doing full transcriptomes. And they have recently changed their pipeline to include spaghettis, because what they used to do is they used to tag either the plasma membrane or the nuclear membrane with GFPs, and then either pull down whole cells or pop the cells and pull down the nuclei. When you use spaghettis, you get a 4-fold enrichment in transcript pull down and something like a 10-fold increase in purity, because you can wash much more stringently. So, I don't know what animal you're in, but we can send you flies, and with Jeremy Nathans, we're making the mouse to do this. Fact sorting is not good. No, I'm serious. These guys, the first thing they did is they said, we're going to do all the technology side by side, and we're going to do the same experiment. Fact sorting was by far the worst sample preparation, because you're shattering your cells and you're getting the background reads in there were horrible. It made it hard to assemble believable data. So, for both fly and mouse, Janelia does manual dissection, I hate to tell you, but you get an order of magnitude improvement in the quality of your reads. And so, huh?
Unless you need to grow the cells.
Yeah, so, but what this does, labeling your membranes or the nuclei, it depends on where your transcript is. I mean, not all transcripts like to live in the nucleus, but if yours does, nuclei are so easy. You can just pull it down with magnetic beads. So the magnetic bead enrichment was something like 20 times better than the fax enrichment. I mean, the fax, you're just really shooting yourself in the foot. Everything's blowing up, and you're getting all kinds of junk. Let me leave you. Oh, just one more fun story.
Question?
Yeah.
Is the fact sorting as bad for non-neurons because neurons have ribosomes all over the place and say microglia don't?
I don't know. At Janelia,
Say shattering your cells.
people don't know what glia are at Janelia, so we only do neurons. Maybe you guys might be more fortunate, but it's a pretty brutal process. Yeah.
Can you finish that? Sorry. I was wondering, can you tag ligand? Because like hedgehog, where they are, it's just pain ass to find out.
Yeah.
Would you tag ligand and then name for that? Would that
Yeah. I thought that's what I showed.
You should a ligand?
Yeah. I mean, you tag cadherins, and then
That's on the membrane? Or
yeah, cadherin are membrane proteins.
no, secreted?
Oh, I don't know if we've tagged as secreted.
That'd be
We
really-
have.
Oh, really?
We have, but it's in some weird fungus. But it worked well.
Oh, really?
It worked well. Yeah. Yeah. Yeah.
So can you do other ions other than calcium? Or is it so boring about
Okay. We'll just settle this right now. Potassium and sodium and chloride, forget it. Just put those from your mind and figure out a different way to do it. Right? I mean, right now, what are we on? Super chlomelian? It's embarrassing. I mean, I read the papers, I'm like, oh, I feel so awful. I mean, they're just terrible, right? And I mean, even the dyes for sodium and potassium are pretty bad. I mean, I'm actually trying to build a potassium sensor with Alan Kay at Iowa. I'm not very optimistic, though, but I mean, we got to try to do it, right? Voltage, I don't know. Maybe 5 years. I don't know. We can talk more in the pub, but I want to show off this guy. And so I want to show, okay, let me just show you this because this is really, we're almost done. So this is showing that the sensors play nice with each other. So this is red neurons and green astrocytes. This is Lin Tian, my first postdoc. And here she's shocking them. Here she's dumping on choline. Here she's dumping on ATP. She's done like 50 more drugs. And we can talk in the pub or talk over email. I was a little worried. I was talking to Ball, and I was like, hey, we've got this cultured study. And he's like, well, Ben is going to tear your new asshole about how, like, it's, the glia are all activated, and, like, it means nothing. And I was like, oh, you're probably right. With those Norwegian collaborators, they verified a lot of our cultural study in vivo. And so I'm like, yay, I think she's got magic hands. So, I mean, these are primary. I mean, a cultured cell, you should never do anything with cultured. They're just monsters. This shows, okay, so this is a hex cell overexpressing EAT2 and then cytoplasmic glue sniffer. And we're pulsing it with waves of glue and then wash. And you can see this, this thing can cycle forever. Then you blow its mind with TBOA, and it's like, and then you wash it out, and it recovers over a little bit. We did salicylate as a control because we also work on SLC26 transporters. Watch this space. It didn't do much, and then TBOA just kind of took it out the second time. I think this could be an awesome, like, high-content screen for transporter agonists and antagonists. I mean, these are untapped drug targets, almost exclusively untapped drug targets. That's where we're hitting SLC26, and there, salicylate has a huge effect. And I'm just show you one last thing. So I sent Joe glue sniffer, and he's like, oh, I'll dick around with it. And then like 3 weeks later, he's like, hey, here's a manuscript I've written about traumatic brain injury. And what he did is, so this is synapsin glue sniffer, and these are layer 5. This is normal expression. The neurons are just chilling. Then he fucking wastes their ass with a big laser burn. And what is shocking is you get universally, and this is 100% of trials, the laser burn is there. It really is that red dot. The glutamate excitotoxic wave is always that size. That means something, and we don't know what it is. And then you can see it starts to resolve, over a couple minutes. Then, I don't know what that slide shows. So then he's like, hey, I'll express it under the GFAP promoter and stick it back in the same region and fry it again and see what happens. And bam, you get your same expansion to exactly the same size circle. Glutamate excitotoxicity knows where to go and where to stop. I don't know, blows my mind. And then as time goes on, it starts to chill out, but you get these little clouds, he calls them, at the periphery. And when you repeat it in the presence of PDC, a GLUT blocker, look, you don't really get the big spread, and then you only get tiny, little, look how small it is. It used to be out here, and the clouds pretty much go away. And it also happens if you block P2X. But then the smoking gun is being in Wen Biao's lab. He found a mouse where they had killed all the microglia. And he repeated the experiment by injecting into both wild type and then microglia deleted mice. And you can see that in the control, you get the big spread, and then you get these crazy little clouds. Bam, 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 bam. There's stuffing going on at the periphery. And in the guy with no microglia, there's nothing going on. So microglia are probably responsible for these little clouds at the edge of the excitotoxic death wave. But what are they doing? Obviously they're coming there to help, but how are they helping? What are they doing? And I think this is something we can really dig into. And so let me just give a shout out to Lynn, who I'm not going to show you any data. I'm just going to tell you a new paradigm that she developed, which blows my mind. It's so clever. She's she's at UC Davis Med School, so she can get patient cells. Routine, right? Routine. You get patient cells, and you de-differentiate them back to some, pluripotent stem cells, and then you can make whatever you want. Run of the mill. She gets genomically sequenced patients, and she not only differentiates them to neurons, she differentiates them to like 5 different cell types and then throws them all together. And she did this from healthy and Down syndrome cases, and I'm going to let so she couldn't be here because her kid's sick, but the answer was extraordinarily surprising, and it is not what the current model of Down syndrome is. So you de-differentiate, so I've missed a key point. You de-differentiate them, you integrate Gcamp, Rcamp, glue sniffer, whatever, then you differentiate them. So they're all labeled with all these sensors, and so then you co-culture them, and you can see where the glutamate is going, you can see where the calcium's going, you can add the drugs, and it was a big surprise. All right, special shout outs, Ball. You rock, man. Thanks for getting me into this field. Ball said, I make science fun for him. You make science fun for me, man. Joe, I have never seen some guy as productive as this. I send him these tubes. Sometimes I don't even tell him what it is. And he sends back like these publication quality figures. So the guy thinks he wants to go to med school. I think we need to go get him drunk at the bar. And he's a scientist, and he's a great scientist, and someone needs to snatch him up. Eric Schreider is my right-hand guy. He solved all the protein engineering work was enabled by his crystal structures. Because he used to be a crystallographer, and the reason he's not here is he's at Woods Hole trying to learn about neurons and ion channels. So he's trying to turn himself into a biologist. He helped make all our sensors. He invented Compari. We're trying to do the Compari scheme with glutamate integrators, voltage integrators, etc. We can chat. He, and I am not fucking kidding. I have the slides, and I'll show them to you at the bar. He and I have revolutionized agriculture. I'm not shitting. So I will show you the slides at the bar. So snatch him up. He's on the job market. Lynn, please God, collaborate with her. She has a small lab. She's just getting going. She's like me. She makes tools, but she actually cares about the problems and the diseases. So, you know, if you collaborate with me, I'll be like, yeah, oh great, I'll have that for you tomorrow. Hey, and then, you know, so, you know, so she'll get the job done. It's focused on disease. She was the first one, made Gcamp 3. She did the astrocyte neuron stuff, did the Down syndrome stem cell. Jasper, he is my proudest graduate because he is now not a scientist. He is a master brewer. And he is at Lost Rhino Brewery that I think you will hear more. They're going to be the next Dogfish Head. They're going to be big. And it's because he actually knows microbiology, and he gets the yeast, he isolates yeast strains, and he sequences them. And then he's like, yeah, this one's going to make a great IPA because of this gene. And everyone's like, whoa. You know? And so, I mean, it's just awesome. Gabby made Emios. We're trying to make other things osmium resistant, other fluorescent proteins, HRP, mini-SOG, all these things. Lots of things could be made osmium resistant. Seratum did all the anatomy work, and I didn't even get to, we do enhancers, and I didn't get a chance to talk about that. You know, Eric I talked about, and Jacob made those transporter screens. The Genie project is big, so these guys are the ones you actually have to thank for Gcamp 6 and RCamp 2. Again, yeah, I shouted out to you. Shout out to Jason Kerr, my fish boys. I love fish. Megan Williams, she helped co-develop the spaghettis, and I knew nothing about EM, so I had a lot of people to help get me going. And now I think it's my bedtime.